Okay, so this is to give you an introduction of GENE-SWitCH, the regulatory genome of swine and chicken, functional annotation during development. I'm coordinating this project with the help of Herbia Klock and Camille Benard, also from INRAE and from INRAE Transfer. So now we'd like to know why it's not advancing. Well, the consortium is made of 11 partners that you see shown in the slides. They cover 8 European countries, and we are also connected with direct links for the operations that we do, the ongoing work, with additional animal breeding companies. GENE-SWitCH has an ambitious global aim, which is made of 2 components. We want to deliver underpinning knowledge on the pig and chicken genomes and to enable its translation to the pig and poultry sector. So it's really a step forward, the simple annotation of genomes. We started in July 2019, sorry. So work is ongoing, and as you may have heard in other presentations or events, we are also connected to other H2020 projects and expanding our aims in the frame of Eurofund. You see the website as a reference. So GENE-SWitCH is made of 3 specific and interconnected aims, A, B, and C. The slide is pretty complex, but I invite you to go to see more details on our websites. I will go very briefly on each of them. So A is functional annotations across tissues and developmental stages for both species, spanning early organogenesis, late organogenesis, newborn animals, and adults through existing data mainly. B is for using the functional annotations for precision animal breeding. It's the focus of today. And C is about standardization of data and processes, dissemination and outreach, which is another main pillar of this project. About aim A, what are we doing? Essentially, we want to identify the functional elements that are activated, poised, repressed in the chicken and pig genomes across the 3 developmental stages that you have seen. And the idea is to characterize their dynamics that we call switches and tissue specificity, as well as their patterns of conservation and variation. Of course, it is implicit in the project is an interesting approach of comparative genomics of mammals versus birds. And we want to deliver high quality, richly annotated genome annotation maps for these species that will be available to the whole community. The underlying aim for moving from aim A to aim B is the question, what's the role of functional genomic elements in the determination of the phenotypes of the adult animal? And of course, here we will gain new knowledge, especially of those elements that are active, poised, repressed during development. And of course, the main question as well is what's the relative impact of the genetic variation of these elements on production traits. So we go to aim B, and here I just restrict my words to the main questions or contents, because this is the focus of this webinar and will be illustrated by my colleagues. So here for this aim of precision animal breeding, we had in mind 2 different approaches. One is of the integration of genetic data. The other one is of integration of epigenetic data, which is more explorative. On the left side, the integration of genetic data, the question is, can functional annotations enhance the prediction accuracy of breeding values in commercial populations? And for the right side, we want to provide the basis for future studies focused on better farm management, for example, using lower quality and more sustainable feed. And about aim C is a quite rich pillar of the project as well. It's about standardization of data and processes, dissemination and outreach. We work in a fair way for findable, accessible, interoperable, and reusable data. for coordination, curation, validation, archiving, and so on, together with the European Biological Archives, and of course the FANG data portal, which is our main main data portal for the data. We do extensive dissemination, communication, and training activities to ensure mutual interactions with all stakeholders. This event is one of them. And as I mentioned before, we are part of Eurofang that was established together with other ongoing H2020 projects like Aquafang, Bobareg, Geronimo, and Rumigen to facilitate collaboration and foster more research. So what is impact that we expect from these components that I've illustrated? First, high quality reference annotation maps for the whole research community that will be, I think, a great resource for both academic and applied research. We will deliver cutting-edge research results that will pave the way for further studies and also, we think, strategies. And we think there will be a number of European stakeholders' benefits, an increased understanding of the value of functional genome annotation to face current and future challenges for improving, for achieving sustainable productions. And this is my last slides. I wanted just to remind all of you, for those who come to Rotterdam, to attend also to our talks that I put in here for each project aim. So there will be one for project aim A, which will be a big summary of what we do for the annotation and the analysis so far in pig and chicken. There will be 4 presentations for aim B that you see listed here, and you will hear more today as well. And I wanted also to remind that under aim C, on 3 July, on the 3rd of July, in Rotterdam, there will be a nice event, I think, that is a joint event of GENE-SWitCH with Geronimo that is also focused on chicken and pigs, Bitcoin or genomics, which brings more.